Howdy, our names are Cassandra Fernandez and Alondra Curiel, and today we're going to report our lab report 7. It's Biochemical Test Lab Part 1. Introduction. Biochemical tests are essential tools used in microbiology to identify and differentiate species based on their metabolic characteristics. The indole test determines the organism's ability to degrade tryptophan into indole using the enzyme tryptophanase. The methyl red test identifies bacteria that produce stable acid end products through mixed acid fermentation, while the bogus Proskure test detects organisms that produce acetylene as a neutral end product from glucose fermentation. The citrate test evaluates whether bacteria can utilize citrate as their common carbon source and ammonium ions as a nitrogen source. Materials. For the media, we use tryptophan broth for the endo test, MRVP broth for methyl red and Focus Proskure test, Simon citrate agar slants. And for the reagents, we use Covex reagent, methyl red indicator, BP1 reagent, and BP2 reagent. And for the cultures, we use Escherichia coli, Enterobacter cloacae, unknown gram-negative bacteria. For the equipment and supplies, we use an inoculating loop and sterile needle, the Bunsen burner, test tubes and test tube racks, pipettes, labels, and marker. For the procedure, we started off with the indole test. We labeled and prepared sterile test tubes containing 4 milliliters of tryptophan broth. Then we used the aseptic technique to inoculate each test tube. And then the test tubes are inoculated for 24 to 28 hours. Then on our next class period, we added 0.5 milliliters of Covax reagent to the test tubes. Then we observed the presence or the absence of red ring, which indicated a positive result. For the citrate test, we lightly inoculated Simon citrate agar slants using a needle, and then we incubated it within the temperatures of 35 and 37 degrees Celsius for 18 to 24 hours, and then we observed the development of blue cutter. And then for the methyl red and VP test, we inoculated both broth tubes with the assigned organisms. We incubated the test tubes for 24 to 28 hours. And then in the next class time for the MR with this, which is the methyl red, we added 1 to 2 drops of methyl red to the test tube. And for BP, we used 15 drops of BP1 and then 5 drops of BP2.
Okay, now for the results for the indole test. Now for the E. coli, we see a bright red ring that's on the surface, and this is basically indicative as a positive result. And basically, we interpreted this that E. coli produces tryptophanase, which just signifies that indole is present. For E. cloacae, we see a yellow top. There's no red ring, so we just indicate like this is a negative result, so no indole is produced. And for our unknown gram-negative, again, yellow top, no red ring, so we just and this result is negative, no indole produced. Um, for our methyl red test, for the E. coli, we see a bright red layer on top, which is indicative as a positive result, and we interpreted this that it produces stable acids through mixed acid fermentation. E. cloacae, we see a yellow orange, but there's no red. This is a negative result. And we interpreted this as producing neutral end products. There's no mixed acid fermentation. And for unknown gram-negative, we see a red top layer visible. We indicated this as a positive, which just means it performs mixed acid fermentation. It's kind of similar to E. coli. For the VP test on E. coli, we see a like not really a different color of a band. We see a band, but it's more like it just stays yellowish. It's not very prominent. And so we indicate that as a negative VP. For E. cloacae, we kind of see this dark brownish color at the top. We indicate that as negative as well. And for our unknown, we see the same thing as the E. cloacae, a very brownish band around the top, and we indicated that as negative as well. For our citrate test, our E. coli, the medium remained green, so it can't utilize citrate, so it's a negative result. For our E. cloacae, there's green, but we see a bluish tint to it, so we indicated that as positive, meaning it utilizes citrate. And then for our unknown gram negative, it completely turned blue, so we indicate that as a positive result, so it utilizes citrate as a sole carbon source. Now for the conclusion. In this lab, we performed the indole, methyl red, bogus Pascal, and citrate test to identify an unknown gram-negative bacterium. The results were indole negative, methyl red positive, bogus Pascal negative, and citrate positive. This IMBIC pattern suggests that the organism can produce both acidic and neutral end products from glucose fermentation and can use citrate as a carbon source. In conclusion, based on the biochemical reactions observed, the unknown organism demonstrates metabolic traits consistent with members of the Enterobacteriaceae family. Thank you for listening to our lab report video.